here. Kristen? Thought so. Sarah? Tristan. Caleb. Jordan. Antonia, right there. Okay. Deshaun. Okay. Landon's Anthony. Okay. All right. Well, let's get started. I guess take my hat off. In the Father, the Son, the Holy Spirit. Amen. God, grant me the serenity to accept the things I cannot change, courage to change the things I can, wisdom to know the difference. In the Father, the Son, the Holy Spirit. Amen. All right. Okay, here we go. So it's time to start. Moving on to the cells, right? We're moving on to the cells today. So basically, there's these three figures in history that are important for deriving and helping us come to know what's called today the cell theory of life. And basically, I'm going to throw these three names up here, their dates. Robert Hooke, 1665, he was the first person to use the word cellulae. He was looking at cork, so he looked at it under a microscope, and he saw that these little units, these little compartments, he called those cellulae. Matthias Schleiden, he was a German botanist, and he said that plants are actually the result of a community of these little living components, which he called the cells. So Matthias Schleiden said living things, plants, are the results of these living structures, single units that he called cells. And then Theodor Schwann, who was a German scientist, he said that animal tissues are also made out of cells. All right, so these three names, know them, know their little contributions and their dates. Hooke, Cork, first used cells. Schleiden said plants are made out of cells. Schwann said so are animals. Easy. Boom. I'm not as worried about dates, to be honest with you. Okay, I'm more worried about the names and what their contribution was. But anyways, the main point is that what's called cell theory is made of these three principles. All living organisms are composed of one or more cells, and the life processes of metabolism and heredity, in other words, all of those processes that keep the cell, keep you alive and help you hand on what is about you to the next generation, that's heredity, those processes occur within the cells. Second, cells are the smallest living things. At least on the planet Earth, cells are the basic unit of all living things, the cell. And third, cells arise only by the division of a previously existing cell. In other words, this is what philosophy would say: we are all contingent beings. We didn't create ourselves. So I remember a fellow, when I was in graduate school, he said, how about this for a graduate project? Try to create a cell in a test tube. Try to create one. You can't do it. You can't do it. All life must come from a life that came before it. All right. So those are the three components of cell theory. So let's talk about cells. So now we're going to move into a different theme of the course. Another theme so what's the one main theme of the course we've been pounding on? Structure and function, right? That relationship. Now we're going to see a lot about unity and diversity. Unity and diversity. There are things that are common, and we're going to see things that are different. All right. So let's start talking about what are some of the characteristics that are common to all cells. All cells. All cells, as we said on Monday, contain genetic material. All cells. DNA. So is a virus a cell? It is not. It contains, well, it does contain DNA, but all right, we're going to see possibly why. Cytoplasm or cytosol. All cells have some form of stuff, a matrix that fills the inside of this cell. They all have it. By the way, related to lab, what's the cell type that when it's mature does not have DNA? Christina. Red blood cells. Red blood cells. You got no DNA. How'd you get to be that way? I don't know, but hey, stop it. Okay, so the cytoplasm, it contains all the sugars, amino acids, proteins, lipids, etc., that are needed for the daily processes. All this stuff that keeps you alive. Cytoplasm is going to contain a lot of that, huh? And as we heard on Wednesday, it's more like Jell-O than it is water. Maybe Jell-O that's like in between. You know, Jell-O that's kind of like not exactly solid, but it's sort of like that in between when you can slurp it. You know what I'm talking about? Maybe in between. Anyways. All cells have ribosomes. This should make sense. If the cytosol contains all the proteins that are needed for daily processes, that means it needs something that can make those proteins, and proteins that make other proteins are called ribosomes. Bam! Yo, let's keep moving. And a plasma membrane. Insane in the membrane. Insane in the brain. Plasma membrane. You ever heard that song before, Insane in the Membrane? I don't remember who did it. Offspring. There you go. 90s. Hey, I'm getting more. That's only 30 years ago. Okay, so the plasma membrane. Phospholipid bilayer that encloses the cell separates the internal environment from the external environment. And we're going to see that within this plasma membranes are proteins that are responsible, help the inside of the cell communicate with the outside of the cell, and also help maintain that internal environment. So some things need to move in, some things need to move out. Proteins that are embedded in the plasma membrane will help us do that. And a common mechanism of metabolism, glycolysis. Glycolysis. Okay, so 5 things. Genetic material, DNA, cytoplasm, ribosomes, plasma membrane, and a common form of metabolism. Glycolysis. Common to all cells. Sweet. What about diversity? So we're going to distinguish now between prokaryotes and eukaryotes. We did a little bit of this on Monday, but let's go over it again. So, prokaryotes. Remember, there's this concept called domains. There's 3 domains. What's the third domain? Eukaryote. Eukaryote? No, I'm tired today, thanks. Eukaryote. Hey, ba-dum. All right, so prokaryotes are found in archaea and bacterial domains. Prokaryotes are single-celled. They're single-celled organisms. You've seen one cell of a prokaryotic organism, you see the whole thing. We see one cell of Quattro, we only see one cell of Quattro. We don't see all of Quattro. But if Sarah were a prokaryote, if we saw one cell of Sarah, we'd see all of Sarah, okay? Because that's all there is to see, one cell. Prokaryotes lack membrane-bound organelles, like nucleus, like mitochondria, like endoplasmic reticulum, like Golgi apparatus. They don't have them. They are all surrounded by a cell wall. Hence, all prokaryotes have both a plasma membrane and a cell wall. And we said that the DNA is in a specialized region called the nucleoid, right? Not a nucleolus. Us, nucleus, nucleolus, that's about you and us. That's a eukaryotic cell. An example, E. coli. That's an example of a prokaryotic organism. Now notice that there are some of these characteristics, they're not unique to prokaryotic organisms, right? It just means if it is prokaryotic, it's going to have a cell wall. But simply because the organism, the cells have a cell wall, that does not make it a prokaryotic organism. What's a eukaryotic organism that has cells with a cell wall? Plants. How about another one? Invite them to parties. Fungi. Hey, the fungi, yeah. Yo! Who invited that thing? Where have you been growing? You don't want to know. Anyways, all right. So, what's another thing that distinguishes cell walls? Prokaryotes versus eukaryotes. What's another way we can distinguish that? Eukaryotic cell walls are made of? Sophia? That wasn't? All right, take a look at your notes. Oh. Peptidoglycans? Maybe, take a look. Okay, that'll be a way to sort of review. All right. Speaking of review, eukaryotic. Let's do it. Bam. Eukaryotic consists of uni and multicellular organisms. What? It's true. Simply because it's single cell doesn't mean it's a prokaryotic organism. If I see a single celled organism with something, what would tell me it's eukaryotic? Golgi, nucleus, etc. Okay, good. What's everybody's favorite organelle? Mitochondria. Love it. Anyways, okay. They contain specialized membrane-bound structures. We call those organelles. And in eukaryotic cells, the genetic material is organized in the nucleus. That's the largest organelle in the cell by far. Right? Where are you going to put your computer? Biggest room in the place. We talked about how we also have this network of proteins called a cytoskeleton. Cyto means cell, skeleton means support. So we've got this network of proteins that supports this cell, helps it maintain its shape, also helps it move, which is kind of crazy, but cool. And some eukaryotic cells will have cell walls, plants and fungi. Animal cells do not have cell walls. You will need to know that for the next section of the course. Animal cells do not have cell walls. That allows us to be movable. It's one of the things that gives us motion, allows us to be mobile, and allows our cells to have crazy shapes. Examples: fungi, plants, protists, and animals. These are the big kingdoms. in the domain of eukaryote. All right, so there's some overlap between prokaryotes and eukaryotes. There's some overlap there. But there are some characteristics that would distinguish, very clearly distinguish a prokaryotic from a eukaryotic cell. That would be important for you to be able to do that kind of a little exercise. If I had to write down 3 things about this cell, I'm looking at it, that tells me it's prokaryotic, what would I be looking for? Think about that. Okay. We'll stop there for a second. By the way, we're sort of on the we're going to go a little bit fast today. That's what that meant. Did you know what that meant, Marie? What's going on? Okay, so questions, prokaryotes, eukaryotes. What about a monkaryote? Where's a robe? I don't know. Anyways, let's move on. Boom. Cell size. Cell size, yeah. So most cells are relatively small. Why? Because they need to efficiently move necessary substances into the cell and get rid of toxic waste products. So what would be some things that we would want to make sure? What would be some things that this cell is going to want to actually move into the cell here to make sure it gets inside, into the heart? Cam. What? Proteins, okay? Maybe we need to get some amino acids from the outside, if we see them floating around, okay? What's some other things we'd want to get into the middle of this thing? Okay? John. Water. What? Water you, no, water. That's right, water. How about something else? From over here. Give you a hint. Oxygen, do we need that? All think so. Most cells do, some cells don't. Maybe sodium, maybe some potassium, okay? What would be some things that we would want to get out of the cell? Like, get out of here. What would be some things we would, if the cell's doing what cells do, we're always going to make some sort of a waste product, right? What's like the #1 animal waste product that we think about? Yep, Sarah. Carbon dioxide. Yo, that's a good one. Absolutely. Hey, how you doing? All right. So, whoa, what did I do that for? Okay. Here's our little itty bitty little cell. It's very small. Here's our larger cell. As the cell gets larger, we notice that the surface area to the volume, the surface area to volume ratio of as the cell gets larger, notice how that shrinks. Now, the reason that's important is because the only way that the cell can exchange waste products and good products between itself and the external environment is through the plasma membrane. I'm starting to learn zombie, so that's one of my, I'm taking that. ZSL, Zombie is a Second Language. You should check it out. It's a new class being offered here. Zombie is a second language. Or Mask. Mask is a second language. I got that. So someday we can all be dentists, right? Okay, so anyways. I'm smiling. All right, don't know if you are, but. So this is the cell membrane. This is where the exchange that helps make life happen happens. And as the cell gets larger, the ability, the efficiency of the cell to move and exchange things between the inside of the cell and the outside of the cell goes down. Because you got a lot more stuff going on. There's a lot more needs inside. It would be kind of like we used to, back when we used to have athletic events where people attended in person, like to use the example of if we were to compare Heinz Field in Pittsburgh versus our Chuck Noll Stadium out here. And if we put all the people, now Chuck Noll Stadium probably has one bathroom, two bathrooms. You know, you get what I'm saying. And then one little concession stand. Now just imagine if you were to then build Heinz Field with the same number of concession stands and bathroom facilities as we do here at Chuck Noll Field. What's going to happen to the ability of the people to either eat or go to the bathroom? How long's the line going to be? Is it going to be long? It's going to be really long, right? Okay. So in order to accommodate all those that increased need for food, for, we could say, getting nutrients in and eliminating wastes, what do we have to do? We have to build a lot more bathrooms and a lot more concession stands, right? Can't just do with one. So one of the ways that the cell actually facilitates this is by creating all of these, not creating, but then larger cells by necessity have these membrane-bound organelles which help with exchange and help carry out different processes in the cell in a specialized way. This is just by way of showing you that here's our prokaryotic cell, somewhere around 3 microns. That's pretty small. We notice here's a human egg. That's 30 times larger. And then here's a paramecium, right? That's a eukaryotic cell. So in general, eukaryotic cells are going to be a lot larger than prokaryotic cells. Ergo, these eukaryotic cells need organelles. Oh, we just went over all this. So I'll post that, okay? So we're going to talk about some of the organelles that help carry out specific tasks. And then the bottom line is a big one here. Eukaryotic cells, they're simply going to need a lot more energy. They're going to need a lot more energy to carry out these processes. They're going to be transporting and moving lots and lots of molecules in this cell across multiple membranes. And oftentimes that always is going to require the use of ATP. All right, so we just went over, I talked about all this. I noticed there wasn't a lot of writing going on, so I'm going to skip the slide. You can pick that up later. Okay, this is just more sort of helping us to maybe understand why these eukaryotic cells have this network of membrane-bound organelles. Cytosol is in the very center of the cell. It's further away from the cell membrane. We have a much lower cell membrane surface to cytosolic volume ratio, so we have less exchange can happen at that plasma membrane. So then we have this series of membrane-bound structures, and we call those organelles. We're going to see the same thing in animals. When we get to the second half of the class, as animals become more complex and larger in size, what are the specialized structures that we have that carry out particular tasks, specific tasks? We call those our organs, right? So organelles are just little mini organs inside of the cells. Right. So the main process we are going to talk about is, we're going to talk about the synthesis of proteins and how it moves through the series of membrane-bound organelles and makes its trip all the way out here to the plasma membrane. So this is from your book. This is our nucleus. The nucleus is surrounded by that double plasma layer, which is called what? The nuclear envelope. And one of the things that we see is that there is this structure here called the endoplasmic reticulum, which is continuous. It's continuous with the nuclear envelope. There's an opening here. So let's talk about the endoplasmic reticulum. So endo means inside, within. Endo, within. Plasma. The word reticulum means net. So when they first saw this, they said, oh, this looks like a little net that's inside the cell. That's why they call it an endoplasmic reticulum. Isn't that cool? Now there's two varieties of the endoplasmic reticulum, which again we talked about on Monday. There's the rough ER. So let's focus on this. And we said that one of the main things that distinguishes the rough ER from the smooth ER is that the rough ER has ribosomes, which are they common to both prokaryotes and eukaryotes? Yes. What's their function? Synthesize proteins. What are the monomers that make proteins? Amino acids. What's the bond that holds amino acids together? Peptide bond. How are those bonds formed? Dehydration reaction. What kind of bond is that? Peptide bond. Covalent bond. Good. On we go. That's called review. Okay, whoa, what the heck? All right. So the rough ER is the region of the endoplasmic reticulum that has ribosomes So it's the one that looks rough or pebbly, sandpapery. That's why they call it the rough ER. It's the rougher of the two. Okay. Now, when the cell is making a protein, the synthesis of that protein always, that's 3 always, begins in the cytoplasm. Protein synthesis always begins in the cytoplasm. Always. I've said that 7 times now. So you're going to know that. So let's talk about this. We're going to talk about this process real quickly. Simply. It starts out as DNA, right? In here in the nucleus. DNA. DNA opens. Zip. And then we have what's called transcription. Transcription. So the DNA gets transcribed into a complementary message, which is called messenger RNA. So it's transcribed. It's going from one kind of genetic language to another. Right? These are both nucleic acids, right? RNA is a nucleic acid, DNA is a nucleic acid. So transcribed, you go from a spoken into a written language. So right now you are transcribing what I'm saying into some form of English on the paper. Depending on what kind of day I'm having, you might be transcribing my Central Pennsylvania English into whatever form of English you come from. Then the mRNA, the message, goes out here into the cytoplasm, gets transported out here. And then the ribosome grabs a hold of it, takes that message and translates it into a protein. So it translates it. It takes it from being a nucleic acid language into an amino acid language. That's why it's translation. So if you were to then take your notes and translate them, put them into Spanish or German, that would be translation. Okay. All right. So the first step, ribosomes initially. So we're assuming that the mRNA has made it out here to the cytoplasm. Ribosomes grab a hold of it. They begin making the protein. They begin translating that mRNA. Putting those amino acids together. As they're linking them, they're in a certain order. What level of protein structure is that? Primary structure, right? Primary structure is the order of the amino acid, the order in which the amino acids are assembled. Okay. All right. So signal sequence. So what happens is, here's my ribosome. These little circles are amino acids all linked together. Doink, doink, doink. So this is the first, the second, the third, the fourth. Now, on some proteins, there will be a signal sequence that identifies that this protein is going to go somewhere. If this signal sequence is identified, what happens then is that the ribosome, notice it's out here in the cytoplasm, it moves. it says, yo, this goes somewhere else, and it goes, and it moves up to the ER. It binds onto the ER. Then the translation of that protein is completed inside the rough ER. So the ribosomes, the rough ER has ribosomes, not because they're always there, but because the fact that they're there means that there's a protein that's being translated inside that rough ER, and the ribosome has moved and taken it there. You got me? Are we together here? All right. So, if the protein is going to be sent to outside of the cell, like in your pancreas. Who's had breakfast this morning? Okay. So you eat breakfast. Your blood glucose starts to go up, up. Signals your pancreas to release insulin. Well, how does it release insulin? That's a protein, that's a peptide that has to be released by cells. So when the cells in your pancreas that synthesize and store insulin, well, that's going to leave the cell at some point. That would be a signal sequence, and the ribosome would then move to the rough ER, and the insulin would continue to be translated inside the rough ER. Cool. To a lysosome. Lysosome, these are, think of this as like a big vat of Clorox in your body. Lysol, right? Lysol kills germs 99.9%. Well, the lysosome contains enzymes that break down all kinds of stuff. It's inside a membrane-bound structure. Why? So that way your cell doesn't digest itself. That would be a bad day. A vacuole in a plant. Or if it's a protein that's going to be incorporated into the plasma membrane. So any of these kind of proteins, their translation will all begin in the cytoplasm, but then they're going to have a signal sequence, which then tells the ribosome, this goes outside the cell to a lysosome, to a vacuole, or this is incorporated into the plasma membrane. Boom, the ribosome moves, attaches to the rough ER, then the translation is completed in the rough ER. That's the rough ER. Sweet. What about smooth ER? Smooth. That's represented here by the green. Well, obviously, the rough ER lacks ribosomes, right? That's obvious. It synthesizes some things. Carbohydrates and lipids. I know steroid hormones are lipids, but I'm going to distinguish those. It assembles membrane lipids, like phospholipids, puts them together. Stores intracellular calcium. In the skeletal muscle cells, we have a particular region called the sarcoplasmic reticulum. Stores calcium. And it detoxifies foreign substances. If there's something toxic to the cell floating around in there, perhaps it comes into contact with the Golgi apparatus, and the Golgi apparatus sort of detoxifies it, like the bomb squad of your cell. Cut the red wire, not the black, whatever. You know what I'm saying. All right. That's the smooth. So now, let's move on from let's get back to our protein release program. So if a protein is going to be, we're ultimately going to say, hey, this is going to be released by the cell. Once it is translated, we see that it sort of buds off, this membrane sort of seals off into a little membrane-bound structure, which then moves through the cytoplasm to the Golgi apparatus. These are like stacks of membranes. That's this yellow thing here. Now it's got two faces. It has a cis face. Cis means with. The cis face is the side of the Golgi that's facing the ER. So that's where these molecules, when they leave either the rough ER or even the smooth ER, they're going to move towards the Golgi. They will then enter into the Golgi through the cis face. And the Golgi apparatus is the post office. Used to be more applicable. The post office of the cell. And its job is to collect, package, and send these molecules to the proper location on the cell where they're needed. So each of those molecules that's made has a particular little signal to it that identifies it as going here, going there. And the location could be inside the cell, outside the cell. Doesn't matter. The Golgi apparatus, its job is to repackage that and send it where it goes. So, working. So, here's the ER, smooth or rough. All these molecules are going to enter the Golgi apparatus through the cis face. That's the face that's closest to the ER. It's going to pass through the Golgi, boom, 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 and then it may get modified. Oftentimes these molecules are modified along the way. They might be changed in some way. They might cut a piece off.
That's only protein too. Carbs can go through there.
Yep, other molecules go through here too, yes. Yep. So if something needs to be finished, this is like the finishing station as well for a lot of molecules. Okay, good question, John. And then it leaves the Golgi through the transface. So the transface is the face or the side of the Golgi that's sort of looking at the plasma membrane, if you will. Proteins and lipids that are synthesized in the ER, yep, they get transported here to the cis face. And here they get modified. The chains something might be cut off. Sometimes other things are added. Sometimes sugar chains or proteins can be added to the lipids, for example. And then the finished packages, then molecules are bound in a plasma membrane. And then they have a particular signal on that plasma membrane that tells these other little proteins in the cell where to take it. This is a very complex system. It's kind of amazing it all works. All this is happening in your cells, man. Like right now as you're sitting here, Stuff is getting made, modified, sorted, shipped, transported to all different parts of your cell, all throughout your body. It's crazy. All right. So, for Friday, that sort of ends. We're going to finish talking about lysosomes, digesting things. And then we are going to move into talking about the plasma membrane. The plasma membrane. So we are moving along. We're going to start talking about the plasma membrane. Let's see. Oh yeah. We have a quiz on Monday, and then we have a test in what? 2 weeks from Friday. Right? Okay, so we're moving along. Remember to review your questions. Review, review, review. Keep going over the old questions. Don't just keep moving forward. Review. Review your functional groups. Review polar, non-polar. Review the different kind of bonds you can form. Keep going over all that stuff. Yo, we're out of here. And if we could have some cleanup help, I think the only things we have left are over there on that side.